I'm just trying to see if I can place myself here so that when I upload the video, it becomes much better. Okay, you all can see the screen, right? Okay, thank you. So, welcome to week two of NPTEL course, which is Proteins and Gel-Based Proteomics. This NPTEL course has the code of BT57. So, in this week two, we'll be majorly dealing with gel-based proteomics and sample preparation for gel-based proteomics. So, what we'll be dealing with in week two is, so before going to that, I don't know how many of you all were there in week one. So in week one, we did discussed about basics of amino acid, basics of proteins, how protein structures are, if there is some misfolding happening, like there are how different experiments were conducted for misfolding and folding experiments, and how important a properly folded protein is required for it to be functional. And then in the end, we did a bit of sample preparation. I think so, or it was about proteins, protein structure, and what was the fifth part of the lecture? If the people who were present, can you all remind me? It was a bit of proteomics, I think so, introduction to proteomics, if I'm not wrong. Okay, so that was about week one. In week two, Sir also gave a bit of introduction about system biology. So, although it is not exactly, it, it's a very broad umbrella. Basically, in that broad umbrella, proteomics is a small part of it. In that proteomics, also gel-based proteomics is even smaller part of that. So we'll be learning that broad umbrella, a bit of introduction about that in part one of this lecture. Then we'll deal with what's proteomics is again, a broad overview of proteomics. Then in that, as we are dealing with gel-based proteomics only. So for that specific type of proteomics, we'll be dealing with how samples are prepared, how protein is extracted, how can we quantify that protein. And after that, one of the technique which is one-dimensional electrophoresis. So we did do a bit of introduction about two-dimensional electrophoresis, if I if I'm not wrong. So in two-dimensional, we do first isoelectric focusing and then the SGS page, right? So in this one-dimensional, we'll be dealing with SGS page only. Like how do we exactly run that protein gel? How do we prepare that gel? How can we analyze results after doing SGS page, etc. Okay, so that's about week two. So I'll go if you have if you all have attended my first lecture. So what I do is I basically do uh, give an overview of the whole five lectures within an hour or so, and then we'll deal with question and answer session. Okay, should we go ahead, or do you all have any kind of doubts in your from your previous week's lecture or the assignment which you all might have submitted? Do you all want to ask any kind of doubts related to that, or we can go ahead with week two lecture? Yes, Ayushi, Himani, Veena, and there's one more, Shrey. So I suppose you meant I can go ahead with the lecture. How about others? Okay. So coming to introduction of system biology, does anybody know what system biology is? Like, can anybody define what system biology is? Have you all heard about that term anytime? The people who can unmute, it would be much better so that we can talk, interact, and then go ahead with the lecture. If it is not possible for you all to unmute, that's fine. Like, at least write it in the chat box. Yes, people. Have you all heard about the term system biology anytime? No? You can say yes, no, at least yes, or else just give a guess like what it might be. Okay. How about others? Okay, you have heard about it. Can you define it if possible, or else just heard about it and doesn't know exactly what it is? So basically, what system biology is, so you're understanding and the whole system. For example, our human body itself, there are n number of systems present in it. So if you do a whole study of human system itself, that's a part of uh, system biology. So basically, you're dealing with each and every section of a system, and you're trying to study as a holistic approach. That's what system biology deals with. So proteomics is smaller part of that system biology. So if we talk in terms of omics, when you're dealing with DNA, it's genomics. When you're dealing with RNA, it's transcriptomics. If you are studying protein, it's proteomics. If you are studying metabolite, it's metabolomics. In that also there is global and cellular. So if you are studying as a whole, it's called as global transcriptomics or global proteomics. Like whatever sample you have extracted, the whole thing you are trying to study, then that's called as global way of understanding omics. And cellular is one specific type of a cell. You're taking that and trying to understand the cellular RNA or cellular proteomic level. That's called as cellular proteomics or cellular transcriptomics, etc. So, giving what proteomics is all about, it's basically a set of all the proteins expressed by a genome, and you are studying that all those proteins by different analysis, different techniques, etc. If you're doing that, that's called as proteomics. So in proteomics, we are studying nothing but the proteome. What is that proteome? Proteome is set of all proteins. So it's a study of set of all proteins encoded by the genome for their expression, localization, interaction, post-translation modification, everything about protein, whatever it is doing, ex- all those functions. So in our first lecture, we did talk about uh, the classification of proteins. Like it can be structural, it can be functional, it can be it is responsible for interacting with something, everything. So if you're trying to study holistic approach of that protein is called as proteomics. But where does system biology come into picture? We'll come to that. So before that, before proteomics came into existence, so genomics was the one which was initially being deduced by the researchers. Like people tried to understand genomics first. People didn't know how to do proteomics analysis. We did do an introductory part of proteomics. It came way later, where the time point at which mass spec came into picture, people tried to understand proteomics in a much better way as compared to that of earlier methods. So genomics was possible because the Sanger sequence, the sequencing of DNA was done very much earlier in 1990s itself. And so although this is blur, so bacterial genome sequence, the whole bacterial genome sequence was sequenced within 1998 itself, whereas human genome was completed in the year 2000s or something. So people did know about how can we sequence a DNA. So by which people try to understand genomics in a holistic approach. But people did not understand how exactly a body is functioning depending upon the DNA sequence, because major part of a DNA is non-coding, right? It's junk DNA, and basically, I think around 98% of it is junk DNA, whereas only 2% of it is coded, and that that specific code of the sequence is translated is transcripted to RNA, and that is translated to protein, which that's how a body or any organism is functional in nature, no matter it is prokaryotic or eukaryotic, no matter how smaller it is, or unicellular or multicellular organism. Although genome codes for something, majority of it is non-coding in nature. Some part is coding in nature, and that has some code to transcribe it and translate it. So by understanding DNA sequence, we did know that how what is the main character which is responsible for the functioning, but how exactly, what type of sequence is uh, responsible for that function, or how is that exactly produced? Are there any modification taking place after the code is being reduced, like after transcription or after translation? Like if there any modification taking place, how it is becoming functional in nature? All those we couldn't understand just by doing genomics, but after that, people tried to understand transcriptomics as well as dealt with proteins to understand in a whole way. That's proteomics. So before that, like it's a very confusing part because we are also talking about system biology and proteomics together. So I'll first talk about system biology initially, and then we can go in detail about proteomics. So what is system biology? As I tried to make you understand about what system biology is in an holistic way. So that it's basically examination of the whole biological entity, no matter. So in that whole biological entity, for let's take an example of us itself. So we are biological entity. In our body itself, there are various systems present, like respiratory system, cardiovascular system, immunological system, everything. And in that each system also uh, there is some cellular interaction taking place, or it is interacting with some other cell itself, or there is some specific molecule inside the cell, like maybe DNA, protein, or RNA, whatever might it may be. It is responsible for interacting with something else. So it is basically an integrated system. Everything has a role to be functional in some other part of the organ, or that specific organ, if it is functioning like this, it is affecting some other system of the body. Everything is integrated basically, and studying that is a bit difficult task. So you're basically examining the whole biological entity as an integrated system rather than studying individual characteristics. So currently what proteomics is doing is it is studying individual characteristics, that is only protein. And in that protein also, everything about protein, like expression, localization, post-translation modification, everything. Although even that specific individual character has subtopics or sub-characteristics of the same. But what do you mean by this specific sentences? Rather than studying one specific individual character, we are studying the whole system as an integrated system. That's what system biology is all about. In that system biology, we are just dealing with one specific character, that is protein, and that is nothing but proteomics. If you are dealing with only genome, that's genomics. If RNA, then transcriptomics. Then if you are studying the interaction or signaling between each and everything, that's called a signaling networks. Then, if you are studying about metabolites, then it's metabolic networks. In that protein itself, if you are trying to study the localization inside the cell, because each cell has different organelles, right? Inside the cell itself. So, inside the cell, each and every organelle has different protein content in it, and different types of proteins as well. Like in some organelle, there will be one specific type of protein which is not present in some other organelle. So all those type of studies can be a part of protein localization. So it is an integrated system. Like if you are studying it as an holistic approach, for example, one protein, it is coded by one genome. Then it will have some RNA, but it is interacting with some other protein, some other uh, metabolite or something. So if you are studying each and every single uh, part of that whole one specific gene, or one specific protein, whatever, that's if you're studying the in an holistic approach, that is a part of system biology. And it's kind of little difficult. Studying one specific character is also difficult though, but that is comparatively easier what we are dealing with now currently in terms of proteomics. But doing a system biological approach is a task to do it, but people try to understand those characters in a integrated fashion. That's nothing but system biology. So may it be respiratory, gastrointestinal. Inside that one system, it can be intercellular, intracellular, or all of these because of one specific molecule. It can be disease, it can be functioning of it, whatever you're trying to study. So these are different levels of system biology, studying one specific system or holistically about each of the whole human body system or whatever, or one specific organism, may it be anything, but those have different levels of it. So this is just an example of each and every system. So why do we need? Because in an individual, everything is interactive in nature. If you are studying one protein, okay, you are studying that one specific protein, that proteomics is there, but where it is coming from, how it is functional, where it is going, and why is it essential to be present in order to be have certain other activity which is taking place inside the cell, that we all are not understanding if you are just dealing with proteomics, for example. That all of those things will be deduced or will be understandable only when you take this specific approach of system biology. That is the reason why we need system biology as a concept to understand each and every, for example, for one specific molecule itself, to understand the whole idea of that specific molecule. You need to have just a minute. I need to have the charging here. Okay. So in a nucleus itself, there is gene, then that is interacting with the transcription factor, that is interacting with the intercellular signaling, which is dealing with proteomics. Then the same part is also interacting with the transcriptional factors again. For example, one specific protein, such as RNA polymerase, for example, that's a protein, right? But it is responsible for synthesizing RNA. For example, DNA polymerase, that is responsible for uh, synthesizing DNA or replication of DNA. So they are all interacting with each other, right? So if you are not studying it in a interactive fashion, studying one specific molecule will not give you any good conclusion out of it. So having a system biology approach will give you some conclusion at a longer stage. So that's what the aim of system biology is: understanding biology in a holistic way rather than reductionist approach. And you are basically quantitate the qualitative biological data. You are quantitating it, whatever qualitative protein, DNA, RNA you are having. And this basically makes a biology a predictive science. If this is happening, then this might happen, or if that is happening, that might be the specific reason because of which this is happening, etc. So all this can be possible when you take your study in a system biological way. So it's like you do an experiment, you find something, that same thing, you are having some kind of an hypothesis, you did an experiment, you have the data, then with the help of data people construct different models, so that specific thing is whatever your experiment you are doing, that specific thing giving you certain result is relevant to one specific objective which you might be having in the experiment. But that same thing, if it is interactive in nature, you are using that specific data, you create models, you do those model analysis and try to interpret that if this is happening in such kind of a experimental setup, it might be happening in other setup as well. Like the same, uh, what do I mean? For example, if something is directly proportional to something, or something is indirectly proportional to something, that you did in terms of one specific objective or one specific molecule, but that is might also be responsible for some other molecule or some other type of an experiment, then that can be predicted by generating certain models and analyzing those models. Then by doing that, you by doing model analysis, you will get end up certain you will end up getting certain answers or conclusion out of it. So basically, that needs to be proven again by doing a certain experiment. So that's how the cycle goes on in terms of system biology. So it can be model based or data based, as we just tried to understand this. So experimental is data based. Basically, you are trying to do certain experiment, trying to collect the data, etc., and then doing analysis how what might be the reason why this got upregulated or this got downregulated, that specific molecule or specific metabolite, etc. In terms of model based, there is prior information. So whatever experiments have been done till now, you have certain data with you. You take up all those data, try to integrate all those data together, because each and every researcher or each and every person might have done that specific experiment, taking one specific sample, or might be having certain objective, and he might have looked at only that specific objective by taking that data and analyzed his data. But that same data might have some other answers for some other questions. So you can take that specific data, create certain model, do certain computational modeling, and try and come up with some questions which can be answered by using that data, create a model out of it, and do analysis of the same. You will get you will end up getting some conclusions out of that. Those needs to be proven again by doing a certain experiment. So that's how the cycle goes on. So studying system components, like as we studied, there are different levels of respiratory, intracellular, intercellular, and the molecular, etc. We just did that slide. So there are different levels for system biology. In that system biology itself, if you are dealing with molecular level, like just the DNA, genomics, just the protein, proteomics, this is interactomics, like DNA binding with protein or protein binding with lipid. That's interactomics. So all of this comes under same umbrella, which is system biology. And different approach for studying system biology. This is interactomics, you can say, like disintegrating the system into its components first, like each and every cellular organelle, dealing with how is it interacting with other organelle, how is so this whole system, for example, consider it as a cell. It's just an example, though you can take it as in any way. So in terms of reductionist approach, you're dealing with one specific organelle first, how it is interacting with other organelle, how it is interacting with some other organelle, how this is interacting with some other organelle, etc. in the whole system itself. If you want a broader example, like for example, this is circulatory system, this is immunological system. For example, this is cardiovascular system, how they are interacting with each other, how it is helpful for to one specific system to be functional. So it depends. Like it's a uh, depends what level you are trying to study and what you are trying to understand in a system biological way of understanding that specific concept. So this can be called as system biological triangle, where below there are the molecular level, then the intercellular level itself, where each cell will have its own metabolic pathway, uh, have its own regulatory motifs present. Intracellularly, that's whole functional module, and in a large scale, the whole system itself. For example, you can relate it in a different way. For example, I'll give you, for example, large scale is is our whole system itself. Functional modules, different organs. In that organ, each and every organ has different organelles in it, and each and every organelle has all of these components inside it. Or else you can just take the cell itself. Inside the cell, there are different organelles, different organelles. There are each and every section of the cell has different regulatory motives, and inside that there are different. For example, this is in the nucleus, this is in the cytoplasm, etc. So it depends how do you want to use this specific system information. This is what we just dealt with, like it. So basically they are interacting with each and everything. DNA becoming RNA, RNA to protein. They are responsible for expression profile, like DNA, how the binding is taking place. So basically, one of the approach is by extraction and mining of complex and quantitative biological data. So this is how complex it is, and we are trying to quantitate it. Integration and analysis of data for development of mechanistic mathematical computational models. So you are integrating by analyzing the data itself, may it be in a mathematical way, computational way, or a mechanistic way, doing certain experiments, etc. And validating those models by refining and retesting. This is basically a different way of explaining the same thing which we were discussing, the cycle of system biology. So this is the same cycle which we just discussed. There's the model, it is will predict something. You need to prove it by experiment, whatever technology you are having, trying to do those experiments, and theoretically, whatever you are getting, is it relevant in an experimental way also? Or else, if you have some experimental data, ha- take those data, do certain computation modeling, predict something else, prove that by experiment, etc. All this goes on. So that's nothing but system biology triangle. So different approach to generate data, going through that cycle, and basically, what's the end goal? Trying to understand the whole system. In that also, like life scientists generally do such kind of analysis, like doing certain experiment, doing genetic modification, etc., and trying to understand whatever you're trying to do in terms of whatever hypothesis you are generating, are they true or false by doing certain experiments. That is a part of life sciences. This is, for example, some who have done bioinformatics study, taking the database, trying to create models out of it, visualizing the whole data and trying to understand if they are having some relation with each other, or the whole modeling concept to make those models, etc. Analyze it and trying to understand the whole system. Those are system sciences. So all this, if we do an interactive, like interdisciplinary approach of studying each and everything, then understanding system biology becomes easier. So it requires data sets from different approaches, from genome, from protein, from transcriptomics, from experimental validation, etc. It requires from each and every aspect of the system biology so that understanding becomes easier. So as we just said, it requires physics, it requires engineering, it requires chemistry, it requires computational science, it requires mathematics, everything. So for studying the whole system, this interdisciplinary approach is required. That's what system biology is, studying each and everything. In that, we are just dealing with proteomics. So it's extremely challenging, and understanding the dynamics of the whole biological networks, it requires modeling, simulation, understanding the whole biology. And in order to do this, there is some mathematical and statistical approach you need to deal with. In that, we are just dealing with proteomics, which is useful to understand the signaling networks with respect to protein only. We are not dealing with signal networks with metabolites or transcripts, etc. So that's the whole summary. So I'll go to proteomics. So that's the holistic way of understanding whole system. In that we are dealing with only protein, like studying only protein or set of all proteins expressed by the genome, wherever it is expression, localizing, interaction, whatever it is. If we're studying that, that's nothing but proteomics, and we can study that using different methods, that is gel-based methods or gel-free methods, using mass spec, using interactomics, or in that protein itself, we just want to study the structure, then these are certain techniques. So in this course, we are just dealing with a gel-based method of detecting or understanding proteins. So that is either SGS page, two-dimensional electrophoresis, differential gel electrophoresis, or native page, etc. So for doing this page analysis, there are different staining dyes available. massive blue, silver, fluorescently labeled stains, etc. So we'll be trying to understand each and every type of stain and each and every type of method which can be used for understanding proteins. So in this course, although we'll be dealing with just these, there are other methods also for studying proteomics, basically to study proteins, like gel-free methods. These are few methods. There are mass spec based methods. Although we'll touch a bit of it about what mass spec is all about and how mass spec helps in analyzing proteins. So there are different ionization sources which helps to fragment the protein. Then those fragmented proteins are analyzed by using mass analyzers. So there are different types of mass analyzers: quadrupole, ion trap, time of flight, etc. Then if you want to study how protein is interacting with DNA or lipid or basically interactomics, so these are certain ways by which you can understand uh, different techniques by which you determine the interaction between proteins with some other molecule or with the protein itself. And if you want to study the structure of the protein, extracrystallography is something which is used to understand the structure of protein. Along with that, there are other techniques as well, like NMR, electron crystallography, etc. So there are different ways of studying proteins, but we are dealing with only gel-based one. So no matter what type of sample it is, blood sample, tissue sample, or just the cell, you can do those gel-based analysis, or you can do microarray-based analysis, etc., and try to analyze those data, recognize the pattern, and try to understand what type of protein or what might be the function of that protein, etc. So, we can either do gel-free method like mass spectrometry, we can do a gel-based method like two-dimensional electrophoresis. To just have the purified protein, you can do protein microarray, or even with the lysate, just expressing the protein using that lysate and trying to understand if your protein of interest is present in the sample or not. You can do surface plasmon resonance as well, wherein here this is called as label-free detection. Although in all of these techniques, like these techniques, you require either staining dye or some label basically to visualize it. Whereas here, in terms of label-free technique, if protein is getting binded, if your protein of interest is bound with the uh, analysis technique you are using, then the reflection, if it is not binding, the reflection stays like this. If it is binding, then the angle changes, and that's how you can determine your protein is present in your sample or not. That's label-free detection. So that's what proteomics introduction is, and sir also wanted to give a broader picture. All those comes under a broader umbrella called as system biology, and he gave certain references here. You all can read about like in detail about system biology using these references. Then in part two, sir discussed about sample preparation. Before going ahead, I hope it's going well. It's a complicated topic about system biology, but here we just deal with the introduction of it because it is very complex because you're studying each and every section of one specific system. Then that's what is called as system biology. Yes, people, can we go ahead with sample preparation? I think I took a lot of time trying to make you understand what system biology is. But now we'll be dealing with gel-based proteomics itself. Like if you have a protein in your sample or in an organism, how do we exactly extract it? What are different types of techniques available to do it? Then after that, basically sample processing in an initial stage before jumping into isoelectric focusing or SGS etc. Are you all there? Give some interact like thumbs up or something like that so that I can go ahead. Yes, people. Yes. Okay. So what do you need to do in terms of extracting a protein or checking whether your sample has uh, protein or not? The first thing is you need so whatever you are trying to do is trying to check a protein inside a cell or inside an organism, right? So inside an organism itself, those proteins are present inside the cell. So first thing you need to do is disrupt the cell. You want that protein to come out. So that's the first step, cell disruption or cell lysis. Then as soon as it comes out, you need to protect it from proteolysis. What do you mean by proteolysis? As the word itself tells you, lysis of the protein, like degradation of the protein. So you need to prevent the degradation, protect the degradation of protein. That's what proteolysis is. So as soon as so inside the cell, a protein is present in an good environment and it is stable inside the cell. But as soon as it comes out, it is not stable in nature. It requires certain pH to be stable. It requires all those degrading enzymes of those proteins to be not to be active so that it is not getting degraded. It needs a chilled environment. Like if the temperature is greater, then it might get degraded. So you need to protect from each and everything so that protein is not getting degraded. Then sample fractionation, separation of sample, etc. So how do we do that? So that all those will be discussed in this specific section. So those proteins are can be present from an organism, from a tissue, from a fluid, from cells, etc. And you need to take a sample which is highly reproducible sample, and it is essential for any proteomic application. And what you're trying to study, that depending upon that, so different proteomic application require different approaches for sample preparation. So depending upon your aim, the type of extraction may change. So if you're trying to study global proteomics, you are global indicating the holistic way of understanding the protein inside a cell or inside an organism. No matter what type of protein it is, you just want to understand all the proteins present in the sample. It can be whatever the function of that protein might be. All types of proteins, no matter what it might be leading to or what its function is, whether it is structural protein, whether it is has some functional inside the cell, whether it is related to some disease, or whether it is related to general metabolism, whatever it might be. If you are studying in each and every uh, protein present in the sample, that's global proteomics. Expression proteomic analysis indicating, for example, you have engineered the cell, you are trying to express one type of a protein, whether it is present or not, then that can be a part of expression proteomic analysis. Targeted proteomic analysis, for example, inside the whole cell, you're targeting few proteins and trying to understand whether that specific protein is being expressed inside the cell and trying to quantitate it. So targeted analysis is generally when you know that that specific protein is present in your sample, then you have standards with you, then you will try and correlate that standard sample with your main sample itself and try to check whether it is relating with the standard, and so you will make different dilutions of the standard and that will help you to quantitate your that specific protein present in your sample. So depending upon the aim, your sample preparation will vary. So process typically involves solubilization. So first step is lysis, obviously. After that, you need to solubilize the protein, you need to denature the protein, do reduction of the protein, basically treating your sample by denaturation, reduction, etc. Then additional steps can be added depending upon your aim. That is to improve the quality of protein extracts. But those specific additional steps may lead to loss of selective proteins as well, depending on the type of species or type of proteins you are dealing with. So this protein extraction protocol should ensure that most, if not all, inside the present of inside a cell or its organelle are extracted. So your extraction technique should be in such a way that most of the proteins are extracted out of it, and presence of interfering compounds are minimized, and good protein extract can ensure success of future proteomics experiment. This is common sense. Like obviously, whatever you are trying to study, it should be as pure as possible, just the proteins. Like obviously, it will have some other components also when you just extracted it, because it's not pure, right? You need to purify it if it is, if you want it, and then you can do the proteomic analysis, because inside the cell there is DNA, there is RNA, there are different metabolites present inside it. So everything will be a part of the cell lysate. It will not be just the protein. It will have everything in it. So you need to make sure that all those other components are as minimal as possible, and in your sample, it should be majorly protein itself. So what are the preparation steps? You need to solubilize it first. You need to prevent protein aggregation. You need to denature and reduce all proteins, and remove nucleic acid contamination, remove other contaminants. So what's the starting point? First thing you need to do the literature. How good sample preparation strategy is already known, and that is what is required for your aim. So whatever your aim is, depending upon that, you need to make sure that you are following that same sample preparation procedure. Obviously, you need to remove other contaminants like nucleic acid, salts, etc. And whenever you are preparing samples, after preparing the samples, always store those samples at -20 and avoid repetitive freezing and thawing. So before analysis of the samples, after extraction, whatever is the different method you are trying to use for extracting the protein, once that is done, if you are not doing the analysis right away, store it in -20. And whenever you are ready with your analysis technique and what your approach of studying that, analyzing the protein, then only remove it and thaw it. Repetitive freezing and thawing makes your sample, it may degrade your sample, so you need to avoid that. So importance of good protein preparation, good sample preparation, like most of the proteins should be out. It should be reproducible, and once protocol is optimized, large number of samples can be processed. So initially you need to always deal with fewer samples, try and optimize the method of extraction, of analysis, etc. Once you get the desired protein which you wanted to study, then you can apply that specific protocol to large number of samples. Make sure all contaminants are removed. So protein extraction from so you need to also check with the source material. For example, your protein might be present in the plasma, but you are dealing with serum. So that's what will not help you all to come to a certain conclusion, right? So make sure that your sample which you are taking has your protein of interest. Make sure it is getting solubilized. So you need to use certain solvents accordingly so that your protein is soluble. And so it basically disrupts all known covalent bonds proteins and removes the interfering compounds. That's the ideal sample preparation. So this is the workflow. Lyse the cell, protect it, fractionize it, extract the protein and solubilize it, and remove the contamination, and then quantify it. So why do we need cell lysis? So basically to facilitate effective disruption of cells or tissue. So you require cell lysis for that, so that it effectively disrupts it, lyse it basically. Then to isolate proteins from intact cells. So initially proteins are present inside the cell, right? So you need to isolate them from the injected cell or tissue. And you need to avoid the loss and modification of protein in that process, and try to get all the proteins out of it, and helps to maximize the sample recovery and also maintain the structure of the protein. So what are the steps in terms of cell lysis? Disrupting the cell, protecting it, and solubilizing it. So how do we do that? So there are different ways of disrupting it. So when sample consists of easily lysed cells, example tissue culture cells, blood cells, these are the ones which are easily lysed. So you need to employ a particular subcellular fraction to be analyzed, like osmotic. So suspend cells in hypotonic media, because of that osmotic pressure, it gets disrupted. You can use certain detergents like Triton X, like SDS, etc., in order to lyse the cell. Or you can use certain enzymatic way of degrading the cell, like cellulase, lysozyme, etc., which helps in degradation of the cell membrane and it will lyse the cell. Or you can use the freeze thaw cycle, like heat it, keep it in a cold temperature, heat it again, like do freeze thawing again and again. Because of the pressure, it will burst out and the cell will get disrupted. So these are all a gentle lysis method. You can use harsh lysis method also, like sonication, French pressure, homogenization, manual grinding, etc. So in sonication, there are some ultrasound waves which interact with the cell, which create certain pressure inside the cell and it will get lysed. So these are certain harsh techniques, whereas these are certain gentle techniques, like chemical way of lysing the cell. These are physical way of lysing the cell. So these are just different variations, like gentle ways of lysing. These are vigorous way of lysing the cell. So depending upon the type of cell you are using, for example, there are certain cells which have cell wall as well. So for degrading cell wall, you cannot just use chemical method and just lyse it. It will not lyse it because cell walls are much more thicker and they are much more strong. You cannot use gentle method for lysing them. You need to have certain vigorous method so that all the components from inside comes out. Then as soon as it is coming out, you need to make sure that your buffer where you are doing where you are adding certain of these chemicals for lysis or using certain physical method for lysing. That specific buffer should also have protease inhibitors with it, like cell releases proteases as well, which may result into proteolysis. So you need to inhibit that proteolysis. So you need to add protease inhibitors to minimize that proteolysis. So these are certain examples of protease inhibitors, like phenyl methyl sulfonyl fluoride. They are effective against serine, cysteine proteases. They also inactivate DTT. They like EDTA is the one which can inhibit metalloproteases. Similarly, EGT also inhibit metalloproteases. So you need to add all of those in your buffer so that protein is not getting degraded. Then why fractionation is required? Because it makes possible to isolate groups of protein. So simplify analysis of complex protein mixture. So you need to separate them accordingly. So allows improved resolution when an individual fraction is analyzed, and it provides less crowded 2D maps basically. So in terms of fractionation, these are different methods. You can purify it using affinity chromatography. You just centrifuge it. Whatever subcellular organelles will get pelleted down, and in the suspension you will just have proteins. Or sequential extraction can also be done based on the solubility. So if you have heard about ammonium sulfate extraction, you can do that for concentrating the protein of interest. You can do electrophoresis. So these are different ways of fractionation of the protein, and that will help you. So your so in a lysate, it will have all different types of proteins together. You can just separate it on the basis of different techniques. Like these are few different techniques, and each and every section can be then analyzed separately, which becomes easier, and analysis becomes much more uh, easy to do basically. So, what is the importance of fractionation? It increases proteome coverage. It separates highly abundant proteins. So, so basically, for example, inside a serum, for example, so there is albumin and immunoglobulins in a higher amount as compared to that of all other proteins. So, what will happen if you take the whole sample and do the analysis? Because of these two proteins present in much more higher concentration, all those other protein analysis will be suppressed because the analyst or the instrument which you are using that might only detect those highly abundant proteins much more frequently as compared to that of less abundant proteins. So if you separate them, for example, separate those highly expressing proteins like albumin and immunoglobulin from the serum, then whatever are proteins which are very minimal in like very less expressed, they can be now amplified and try to understand that these minimal proteins. So basically, The analysis of those proteins or detection of those proteins will be much more efficient if you remove that highly abundant proteins. So separating highly abundant proteins from low abundant proteins of interest bring them into dynamic detection range and increases the chances of identifying that low abundance proteins. That was about part two, about initial part of sample preparation. And so this is, for example, serum proteomic analysis, which I just discussed, although we'll discuss again in this part three. How extraction is taking place, how can we concentrate the protein by precipitating, how can we remove the contaminants. So we just did the first part, that is lysing and fractionization. So in that fractionization, after fractionization, for example, you need to concentrate the protein of your interest. So subcellular fractionation to enrich the protein of interest. So there are different ways of doing it, using by desalting or by using TCA like trichloroacetic acid, which helps in protein precipitation. You can use acetone as well for protein precipitation. You can use both of them like TCA and acetone together. So depending upon the protein of interest, depending upon the sample, the source, etc., how much amount it is, if it is very much, very minimal or very abundant in nature, depending upon the uh, type of protein you want to analyze, the extraction method will change. So first is solubilization. So naturally form complex with membranes, nucleic acid or other proteins. That's what proteins generally do, but form various non-specific aggregates as well. So that precipitate when removed from the normal environment, the solubilization becomes easier. So there are different components which can be used for solubilization or denaturation. So for example, urea denaturates it and helps in solubilization. So it basically unfolds the protein and makes it soluble. It helps in solubilization, unfolding the protein, and also gives ionization to it. Like it gives charge to the protein, and that's how solubilization is getting improved. Also, you can use certain detergents like SGS, which is extremely effective in solubilizing hydrophobic proteins, and so it is not compatible with isoelectric focusing. So you cannot use SGS if your further aim of protein analysis is isoelectric focusing. So that's what we were discussing, right? So depending upon your aim, depending upon the type of analysis you want to do, depending upon the type of protein you have, accordingly, the initial sample preparation changes. For example, if you are dealing with IEF, you should not dissolve your protein in SGS first. You can do it in later if you are doing two-dimensional electrophoresis. But in the initial step, you cannot do that because isoelectric focusing is not compatible with it. Similarly, there are some other types of proteins like chaps, which is this, and it is deuterionic and it also gives some charge to it. So basically neutral charge to it, and it helps in isoelectric focusing. You can add certain redundants like dithiazretol or TBP. It is most common, so it removes the disulfide bonds present in the protein. It increases the solubility of protein by giving some non-ionic reducing agent. So depending upon, as we are just saying, like depending upon your protein of interest, the solubilizing agent will differ and that will help you to solubilize the protein. You can use certain ampholytes as well for sample preparation, like for solubilizing the protein. So what does it do? It basically minimizes protein aggregation and enhances protein solubility. So either use this, either use reductant using carrier ampholytes or detergents, depending upon your type of protein. Or some methods might use two or three methods together for completely solubilizing the protein. So typical sample solution. So this is the, like as I just mentioned, you can use two, three together also for. So in a typical sample solution, you have urea, thiourea, you have chaps, you have DTT. So this has a role of uh, breaking disulfide bonds. This has a role of solubilizing the protein by giving certain charge to it. Similarly, chaps, etc. So each and every component has its own role, and overall, it will enhance the solubilization of the protein. So depending upon the type of cell, plant cell, fungal cell, so these are tough cell walls. So it will so proteolysis problem exists, and you need to use some physical way of lysing fungal cells. In terms of bacterial cells, there is high ratio of nucleic acid protein, so you need to make sure that you have a technique which removes DNA with it. DNA, so that it is not, so that protein is not contaminated with nucleic acid. So that's what. Depending upon the type of sample, your sample preparation technique will change. So some pre-analytical factors like aims for simultaneous analysis of thousands of proteins, so which occur prior to the point of actual sample analysis and are biological or technical. So it can be intrinsic influences like gender, age, ethnicity, or extrinsic influences like diet, medication. So depending upon this, a specific individual might have different types of proteins present inside his body. So it can be either intrinsic influence or extrinsic influences. So if you are dealing with a large cohort of patients, you need to make sure that all these factors are present with you, so in order to interpret the data. So technical effects of sample collection, processing, and storage, the gross effect. So all these factors you need to consider, because although you will just collect the sample, you will do everything, but analyzing it will be difficult if you don't know all of these factors, because you will find that in one specific sample, that specific protein was highly upregulated, in one sample it was highly downregulated. But why is that so? Like why in that individual sample we got some proteins to be overexpressed, and that in another individual sample it was downregulated. If you don't know their how they came from and what was that specific individual extrinsic factors, were there any influencing factors of uh, for that like he was he following some specific diet, was he smoking, was he alcoholic, or has certain larger age or very young age, or he belongs to some different ethnicity itself. So all those factors you need to make sure you have all of those with you, so that after sample analysis you can relate it and try to understand why those specific proteins are being found in a high amount or a low amount or a similar amount. So you need to make sure that all those are present with you during your sample collection, and how you are handling the sample. All those will have some effect in your protein analysis. These are technical factors, basically how you are storing it, how you are collecting it, etc. How you are then sample separating it. Like so, in terms of precipitation, as we just mentioned, there is ammonium sulfate method. So due to high salt concentration, protein lose water in there and aggregates. Basically, it precipitates, and so depending upon the concentration, that specific type of protein will get precipitated. So accordingly, fractionation of proteins can be done with the help of different concentration of ammonium sulfate. There is some method called as acetone precipitation, wherein you use acetone for precipitating the protein, which are, so this is for organic solvent soluble contaminants, like it will remove them like detergent, lipids, etc. And the excess of at least 3 or 4 volume of ice cold acetone. So it needs to be done at a colder temperature, this type of precipitation, and it allows protein precipitation. And later it can be pelleted down using centrifugation, and acetone can be removed. And the precipitated part will be having only protein in it. Similarly, there is TCA method also, similar type of method as that of acetone. You can use both of them together too, and allow maximum protein to be precipitated. This is ammonium acetate in methanol. So there are different solvents basically in order to precipitate your protein. Depending upon the type of protein you want to get precipitated, different methods can be applied, and maximum amount of protein can get precipitated. So it will be concentrated also, and analyzing those concentrated protein would be much easier and much lesser sensitive technique can be used because your protein is concentrated. If it is not, it will be present in very minimal concentration, and then you need to use a very specific sensitive technique to analyze those proteins. So that's what it helps in terms of doing protein precipitation. So once that is there, if there are certain contaminants present, you need to remove them. For removing, there are different methods available. So like in terms of high amounts of salts are present in biological samples, you need to remove salt. So these are certain different methods for removing salt: dialysis, gel filtration, precipitation, etc. Out of which dialysis is a popular method. If there is nucleic acid present in your sample, so they bind to proteins through electrostatic interaction. You can prevent proper isolectric focusing and produce severe streaking if you have nucleic acid in your sample. So make sure that you remove that first and then you go ahead with isolectric focusing. If your sample has that, then you will see streaking of the sample because of the presence of nucleic acid, because it forms complex with the carrier ampholyte. So you need to make sure that those are removed, and those can be removed with the help of DNAs, RNAs. You need to add that to your protein sample so that DNA gets degraded, RNA gets degraded. So these are the ways by which you can remove the contaminants. Similarly, polysaccharides, if they are present in your sample, majority of the time they are not present, but if even if they are there, because they are larger molecules, one centrifugation, those things are generally removed, but they can be removed by these precipitation methods. In terms of lipids, if low amount of lipid is present, solubilization should disaggregate lipids, and so basically lipids won't be soluble, right? They are hydrophobic in nature, so generally by doing that uh, solubilization, lipids are generally out, and so you can do chemical delipidization as well prior to resolubilization. So this is a method can be achieved by extraction with organic solvents containing chlorinated solvents or ethanol or acetone. So that will also help you to remove lipids from the protein sample. Then adding detergents can give you negative charge to the protein and separating them. So that's how proteins can get separated out from the other contaminants. These are some other interfering compounds, and so there are techniques available to remove them too. So that is what we discussed in part 3. Then similar to that of methods which helped in sample preparation, there is some method called as Trizol method too, wherein if you are dealing with bacterial sample, this is how the whole procedure is. There is a bacterial sample, you are precipitating the bacteria. Then this is the same thing, how the bacteria grows in a suspension culture. This is in the plate, and this is in the suspension culture. You can just pellet it down, sonicate the sample so that the cells get lysed out. Then after that, we dealt with different solubilization, right? So one of the method is called as trizol method. So once, as soon as you add the trizol to the cells will get further lysed and everything will come out, and it will make sure that DNA, RNA, etc. are not interfering with your protein extraction. Then once you centrifuge, mixture shows separation of proteins, DNA and RNA. That's what trizol do basically, and it will pellet your protein. Once you remove that, there is a procedure basically. It will not remove randomly. So you need to add certain solutions to it, and your protein gets pelleted out. So you can dry that and reconstitute that protein and do the analysis. So this is one way of preparing your samples in terms of bacterial sample. So what you do is you use 2% SGS, give an heat treatment so that it gets lysed. You can add lysozyme so that cell membrane of the bacterial cell gets lysed. Then you can do acetone precipitation, whatever proteins comes out. You can do TC acetone precipitation, whatever protein has came out will get precipitated. You can do extraction of those protein itself by using solubilization buffer and or else by trizol method. The trizol method will separate DNA, RNA, and protein. So, what trizol basically has guanidinium isothiocyanate and is inhibitor of RNase, and it gives good quality RNA. But we want to remove that. We are interested in protein here. So trizol is also used for RNA extraction. So that's what it is written here. So you can, if you are using supernatant, that becomes your RNA extraction, and you can take that for a different procedure for extracting RNA. Then this will help in make sure that there is no nucleic acid contamination, and it will not require any desalting method, which is generally required for ammonium acetone precipitation or TCA precipitation. So for all those methods, you require desalting method also. Desalting step, if you are doing with trizol, you will not require this step, and you will also not require lipid contamination. So all this will be removed for sure if you are using trizol method. And proteins are also easier to resolubilize using this method. So what is the method? Basically, you add 1 ml of trizol, you add 200 microliter of chloroform, you mix it, keep it in a 15, keep it for 15 minutes at room temperature and centrifuge it. Then you need to carefully remove the upper layer which contains RNA. Then the bottom layer, to the bottom layer, you can add 300 microliter of ethanol, centrifuge again. Then ethanol will precipitate the DNA and it will help to remove the DNA. Then the supernatant contains the protein, and that can be further taken to add further taken for extraction of the protein out of it by using chilled acetone. Then after incubation with acetone for 4 hours, basically at minus 20, you do the centrifugation, then discard the supernatant. Then the pellet is something which has the protein. You can wash that protein using ethanol and reconstitute that dried protein in a lysis buffer, and then use for analysis. So that's how the method is in terms of protein extraction. You can quantify that using UV spectrophotometer or by Bradford method, etc., or using BCA method or Lowry assay, whatever method you want to do the analysis. And that's how we can analyze or quantify the protein after the whole extraction. So we studied about how samples can be prepared, what's the protocol for doing, and how do we decide what type of sample preparation is required depending upon the type of protein. And sir has given one example of bacterial preparation. For blood sample, for plasma, for other types of sources, you have a different method. So for bacterial, one of the method is trizol. So once your sample is now prepared with you, now you need to analyze it. So one of the way of analyzing is SGS page or native page. So electrophoresis was basically invented by Prof. Arne Willem in 1930, and he developed this specific method, which is moving boundary method to study electrophoresis of proteins, and it is now widely adopted by 1960s. So this is one-dimensional technique. You are separating the protein in one dimension itself. It basically separates depending upon charge to mass ratio and molecular weight of the protein. That is using sodium dodecyl sulfate, which gives negative charge to the protein. And native native page is nothing but polyacrylamide gel electrophoresis, which is PAGE. So how do we make that specific gel, or how do we use electrophoresis for protein characterization? So there is subunit composition, molecular weight of subunits, because you are using SDS which denatures the protein. In terms of native page, the native molecular weight can be determined, and post-translation modification, if there are anything present, you can characterize them as well using electrophoresis. So it's a single dimension, a large number of proteins can be separated, and complex mixtures like serum, cell lysate, etc., can be separated using this method. So in terms of first one time, so we are dealing with SGS and native page. What does SGS do? So it is widely used method. It gives negative charge to the protein, and boiling proteins in SGS and thiol agent like beta-mercaptoethanol, it denatures the protein and breaks the disulfide bond. And so what's page in that? So there is SGS and there is page. Page is nothing but polyacrylamide. So acrylamide plus bisacrylamide, they are bound together to make a poly structure. So that's polyacrylamide. So this is how it looks, acrylamide and bisacrylamide. And they are joined together to make polyacrylamide, but that happens only with the help of APS and TIMID. APS is the initiator of that polymerization. They both will bind together and make the whole chain out of it, polyacrylamide, with the help of APS, that is initiator of polymerization. And TIMID is the one which promotes the polymerization with the help of free radical stabilizer. And that's with the help of TIMID and APS, a polyacrylamide is made. While solidifying, like initially they are liquid in nature, but uh, when the polymerization takes place, they become solidified. And along with that, we also add SGS to it, which makes the protein negatively charged. And if there are disulfide bonds present in your sample, you can use beta-mercaptan ethanol to break those disulfide bonds. That basically gives negative charge to the protein like this. Once you do the sample analysis, and they will run on the basis of molecular weight, the smaller one will move faster and the larger one will move slower. So it is commonly used for determination of molecular weight of unknown protein along with the protein markers. So you can either use these markers or you have a ladder as well, like ladder like this. It will help you all to determine whether your protein of interest is present or not. So this is how the silver stained gel looks like, and this is how fumassive stained gel looks like. So silver stain is much more sensitive in nature as compared to that of combative blue. So it is an anionic detergent. It gets negative charge, and 1.4 gram of SGS per gram of protein is the binding ratio. And uniform charge to mass ratio is provided by SGS itself, and thus separation is solely on the basis of molecular weight and no charge. And in terms of native page, protein analysis under native page. So here you can, so it depends upon your objective, what you want to understand about the protein. So it provides necessary charge to the protein because of this dye and facilitate the separation in the gel. Unlike SGS, dye does not have denature the protein but binds to them in a native state itself. So, so this helps. So, what is the application of native page? If you are not denaturing it, you are not giving negative charge to the protein. So, it is a multi-protein complexes if they are present. So, they are not degraded, right? They are not denatured. So, if there is a multi-protein complex present in your sample, you can identify that using native page. And so this electrophoretic mobility is determined by the negative charge of the bound comassive dye. So here the negative charge is being given by this dye and not because of SGS, and size and shape of the complex. So depending upon that, the gel will it will run the sample, and it helps you all to subunit. So this is the overall summary of the fifth part of the lecture. You molecular weight of the subunit, native molecular weight, and post-translation modification. If anything is there, you can use electrophoresis and understand that. That's what the sir talked about in his 5 lectures. So I think today it went a bit longer though. So maybe we'll take a break of 2 to 3 minutes only, and then we'll deal with question and answers. So that was the week 2 lectures we sir talked about. I don't know whether you all have gone through those lectures which have been provided to you all. So in this week 2, we are basically dealt with a bit of introductory part of what system biology is about. So where does proteomics is coming into picture from a broader umbrella, which is system biology. In that, a small part of it is proteomics. In that proteomics also, there are different ways of studying it. And in that also we are just studying gel-based proteomics. And in that gel-based proteomics, today we just learned about, depending upon the sample, depending upon your interest, what you are trying to study, your sample preparation will change, your sample collection will accordingly be there. And depending upon the type of protein you want to analyze, you will accordingly extract the proteins and make sure that all those contaminants are not present in your sample and only protein is there. And we discussed about different methods related to that and how that specific protein can be analyzed. In that, we just discussed about one specific method, which is SGS page or native page, basically electrophoresis. That's what the whole overview of week two lecture is all about. We'll take a break of like for 3-4 minutes break and we'll start with question and answers. If you are till then, if you are having any kind of doubts, you can take a break, or else if you have any kind of doubts, you can just yes, Himani, you can write your question or unmute yourself and answer. Sorry, unmute yourself and ask the question. Read this question and try to solve it. Oh, sorry. Yes, are you all able to solve it? Okay, Aparna, how did you solve this? Yes. What's the protein say? Like question says, it has the molecular weight of 400 kilodalton. There's one protein. You have done gel filtration and subjected to SGS, and the protein showed three bands: 180, 160, and 60. So when electrophoresis was performed in presence of SDS and DTT, again three bands were formed, but this time with molecular mass of 160, 90, and 60. So what is the subunit composition of the protein? So basically you need to find that. So there was, so 60 is there. So this 160 and 180 has some interaction with disulfide bonds, right? So, so when the electrophoresis was performed using the presence of SGS and diethyltritol and beta-mercaptan, so these two are responsible for, yes, responsible for breaking disulfide bonds, right? So, so 60 kilodalton is there already, but it showed you 160 and 90. What is the subunit composition of the protein? So this 90 is basically coming from this 180 itself, wherein this specific 180 band might be having disulfide bonds with it, and the breaking of that disulfide bond has taken place with the help of DTT and beta mercaptan, and that's the reason it could find 90 and not 180. And the composition of that would be 60, 90, 90, and 160. Are you all getting it? Others who could not solve this specific, Alpana and Veena Iyer could do it, but I don't know how about others. Are you all there? Yes, Ray, if you are there here, and I don't know who all are present. So there are 10 people, and I could see only answers from two. If you could not solve that, I hope when I try to explain, you all got it. And if not, you can ask the question. So that's what the answer is: 60, 90, 90, and 160. Okay. So in a quantitative proteomics experiment contains large number of samples, which of the following would be preferred choice of method? So there are large number of samples present. So although we don't know about it, just like we have not read about the gel-free based techniques here, but using your common sense, like if there are large number of samples present, what would be the preferred choice of method in order to detect them? Which one would be much most cheaper and most faster way of analyzing uh, and quantitative way as well? Yes, the answer is C. So if you want to do quantification, the answer is in the options itself. You need to quantitate it, and label-free quantification is the preferred choice of doing an experiment. All those would not help you to quantitate it, but qualitatively you can tell whether the protein is present or not. So label-free technique will help you to answer quantitatively. Yeah. So serum contains albumin and globin. There are high abundant proteins make up 80% of the protein content. You're working on a serum sample. You want to identify low abundant proteins. Which of the following method will you use to increase protein coverage? Yes. So we did discuss about this. You need to remove that so that those low abundant proteins are detectable. So you'll be doing depletion of high abundant proteins. Yeah. So you want to study mitochondrial enzymes involved in beta oxidation of fatty acid. Which of the following method will you use to fractionization of subcellular organelles? So you need to make sure that all those subcellular organisms are removed and only mitochondrial. So you are dealing with mitochondrial enzymes only. So you need to have one specific method by which you can just do the analysis of one thing, that is mitochondrial enzymes. So it also gave you greater hint that cellular organelles need to be separated. Yeah, obviously. Good. Yes, try Neha Mondal. Try and participate in this question and answer session. So, which one of the following is not an element of system biology? Biological network, modeling, computation, and pre-fractionation. Great. Yeah, it's the pre-fractionation. So, protein precipitation can be performed by which of the following solvents? Acetone, TCA, acetone, isopropanol, or all of the above. The cross-linking of agent of polyacrylamide gel is it's bisacrylamide. That is what is cross-linking with acrylamide. Timid is the initiator, like it's enhances and APS is the initiator. Okay, this will take time probably. So read the question properly and try and answer. So there are five proteins A to E and listed the molecular weights are being listed. The isoelectric point is also listed. Give the order of migration from top, the point of sample loading to the bottom of the gel. So it's from top to the bottom of the gel. So it's asking about SDS. So everybody says C. So first is E, which is the highest molecular weight, 97,000. And then you say B, which is 83,000, which has a PI of 10.6. And then say C, which has 47,000. Then A, which is 35,000, and D, which is 19,000. So EBCAD, that's the answer. So as the separation is solely on the basis of molecular weight, you don't have to look at isoelectric point here because the question was from top to bottom, wherein the gel runs in terms of SDS page. So this is for making creating confusion here because SDS solely runs on basis of molecular weight. So you just need to look at molecular weight for answering that question. Yeah, so membrane proteins are highly hydrophobic. So for hydrophobic proteins, and they are difficult to extract from the membrane. In light of this, which of the following reagent should be used for solubilization of membrane proteins during extraction? Yes, thiourea. That's the answer. So your protein sample is a lot of phosphorylated proteins. In this slide, which of the following stain would you prefer to use detection of phosphorylated proteins? Oh, we didn't do that though, but you can try and answer it. So it's about phosphorylated proteins. Just make a guess if you want to, but it's a theoretical question. So if there is one specific dye which is helpful in understanding phosphorylated proteins. So it's ProQ diamond, which is the one which is so all this can stay in all types of proteins, but if you are dealing with phosphorylated one, this is the dye which is used for uh, checking phosphorylated proteins. So which of the following sample reagent is added for protein sample to provide charge in blue native page? We discussed this. So what's the answer? It's blue native page and not SDS page. Someone answered SDS. Sorry. Still, you can just read the question. So lipids often interfere with proteomics experiment and removal is essential. Which of this organic solvent is used to remove lipid contamination from protein sample? It's methanol. Which of the following statement is true for SDS page? Proteins are denatured by detergent SDS and boiling of sample. Upon SDS treatment, all proteins possess the same charge to mass ratio. Smaller proteins to migrate more rapidly through the gel, or all of the above. All of them are true, or any one of them it is true. Good. I can see most of you are answering most of the questions correctly. Salts often interfere in two-dimensional electrophoresis experiment and should be removed to avoid clogging in IP district. Which of the following technique is not employed for removing salt? So you are dealing with salt suppression. Which of the technique is not used for salt suppression? Yes, people. Which of the following method is not employed for removing salts from protein sample? Equilibration, gel permeation, chromatography, precipitation, dialysis. So people might be confused between A and B most probably. The answer is A, whereas all other techniques can be used for suppression of salts. Which of the following is an aim of sample pre-fractionation? Pre-fractionation, why it is done? What's the aim of pre-fractionation? Increasing the proteome coverage, reducing sample complexity, removal of high abundant proteins, or all of the above. Which of the following statement is true for off-gel fractionation? So question 16 to 2 are lab session based questions. It's true for off-gel fractionation. Gel side of the IPD strip should be kept upwards. I don't know whether you all gone through that video. I haven't shown that gel lab session based part. I haven't covered that too. So it's there in the videos. If you have gone through it, it will be easier to answer these questions. So it was gel side of IPD strip should be kept upwards is true for of gel fractionation. Heat stabilization technique has significant advantage over the conventional approach like snap freeze because so the all these are lab session based questions. So I haven't gone through that. So that was practical. So I just talked about theoretical uh, concepts in the lecture. But in terms of lab sessions, so there are videos which are being provided. You just need to go through that, and all this would be very easy to answer. So in heat stabilization, they are free of additives, and that have a significant advantage over conventional approaches like snap freeze. On which of the following basis does protein peptide separation occur in off-gel fractionation? Mass, molecular weight, isoelectric point, or none of the above? So off-gel fractionation is nothing but isoelectric point like or IEF technique. Proteomics studies use brain tissues sample are often used to understand the progression of brain tumor. Tissue samples should be handled with care and different strategies are often exploited. Heat stabilization of tissue using denator involves what? So it's the same question of heat stabilization. Involves denaturation of proteins, reduction of enzyme activity, unfolding of proteins, or permanent inactivation of enzymes. So this technique basically halts the enzyme activity. So it's permanently inactivates of enzyme when you use this denature heat stabilization technique, and all those enzymes are inactivated so that your protein analysis can be taken place. Unable to understand the lab session. It was practical, right? Like they have shown the how it is needs to be done, right? Have you gone through the videos? Okay. So you were it was difficult for you to understand the lab session. So if that is the case, what I'll do is like in the next week lecture, I'll first cover that lab session video and try to make you all understand what that lab session is. Okay, when Himani couldn't understand, or else even Veena could not understand exactly what that lab session is. So what I'll do is in the next session, like in the third week lecture, I'll try to cover that lab session first, and then I'll talk about uh, the week three lecture overview. Okay, I can do that then. I thought like it was practical, so it would be easier to understand. I just dealt with the theoretical part. But if that's the case, what I can do is I'll go through that lab session thing and try and revise that first, and then I'll go to week three. Okay? Yeah, I'll do that then. So let's go through the last question once, and then we'll try and go through this session again, like these questions again, like from 16 to 20 to like the next week as well. So biological samples are extremely complex and contain different biomolecules and small metabolites. The main role of gel fractionation in case of biological sample is to the main gel role of gel fractionation in case of biological samples is to reduce the complexity, dissolve protein peptide on the basis of the PI, and separate proteins on the basis of hydrophobicity, or both A and B. The answer is both A and B. So this answer you might be understanding by this question itself. Isoelectric point of so this is the protein occur in off-gel fractionation. So obviously this will be there like resolving proteins peptide on the basis of pi, but it also reduces the complexity. So answer is A and B. So what I'll do is in the next lecture I'll go through this lab session talk first, and then we'll talk about week three lectures. So despite that, do you all have any doubts? I think one of the student also asked me to do SPR, and what was the other technique? Like I think I have a chat with me, but I'm not remembering it. It's one was SPR technique and one was IP something. I'm not remembering it. So those two techniques as well as the lab session, I'll try to include in the week three lecture. Are you there? The one who asked me in the first lecture? I don't know who asked me also. Alpan asked me. Okay. Biolayer interference. I'll just note that down. So biolayer interference and SPR, right? Okay. So I'll go through this. I'll go through SPR and the lab session video of week two. so that it becomes easier for you all to understand the topic. Anything else you all want to ask me, or you all want me to include in the week 3 lecture? Yes, I hope it's going well. Like, I don't know if you all want anything else with this TA session. Okay, I need to go through this technique. I don't know exactly what is this technique. I do know about SPR. I can explain about SPR, but I need to go through this technique, uh, biolayer interferometry, and I can accordingly explain. I have never done that or never heard about. Basically, to be frank, I've never heard about this technique too. But I'll try to go through that if I can relate with that specific technique and try and understand it, and I'll explain it. Okay, anything else? Nothing. Okay, then we can leave the lecture. If someone wants to ask anything, they can ask. Okay, then thank you. Thank you for attending the class. Next week, I'll try and cover all those doubts. Thank you.